I'm Richard Hodis, Director of the National Institute on Aging, and it's my pleasure to welcome you to the Florence Mahoney Lecture, Understanding and Modeling Aging, that will be presented by Dr. Anne Brunet. The NIH Mahoney Lectures are part of the NIH Directors Series and are named to honor Florence Stephenson Mahoney, who, until she died in 2002 at the age of 103, was enormously accomplished, an exceptional woman who devoted much of her life to advocating for the creation of NIA and for increased support to NIH. And I would meet each year at her home in Georgetown to discuss with her our plans for the upcoming Mahoney Lecture. She was sharp, she was strict about the kind of science she wanted to hear, and I know she would be thrilled with Dr. Brunet's presentation upcoming. Anne was scheduled to present last year, but that was postponed due to the COVID-19 pandemic, and we're very grateful she was able to reschedule. So I'm now honored to introduce Dr. Anne Brunet. She's an NIA grantee. She is the Michele and Timothy Barakett Professor of Genetics, co-director of the Paul F. Glenn Laboratories for the Biology of Aging at Stanford. She earned her BS degree with honors in biology from the École Normale Supérieure in France, her PhD in biology from the University of Nice. In 1997, she moved to the United States to pursue a postdoctoral fellowship at Harvard, then to Stanford in 2004. She has received numerous awards from NIH, including the NIH Director's Pioneer Award, the Bennett Cohen Award for Aging Research, the Michele and Timothy Barakett Endowed Professorship, and the NIH Director's Transformative Research Award, all in acknowledgement of her extraordinary accomplishments. She's been interested in the molecular basis for aging with particular emphasis on the nervous system. Her lab is focused on understanding aging, the potential for rejuvenation, tackling the complexity of aging and longevity, and a lab using multiple model organisms, diverse lifespans, worms and mice, but also the killifish, which she's really taken as a unique addition to this list of informative models. Her lab is currently interested in identifying the mechanisms underlying aging and the pathways involved in regulating aging in response to external stimuli. They try to understand the mechanism of brain aging and rejuvenation, focusing on the role of regenerative neural stem cells. I'm delighted then she could join us today. Join me in a warm welcome to Dr. Anne Brunet. Anne.
All right. Thank you so much, Dr. Hodis, for this very nice invitation to come to NIH virtually and NIA virtually. And also thank you so much for this very nice introduction. Good afternoon, everyone. It's a pleasure to be with you guys in a virtual manner. So as Dr. Hodis mentioned, our lab is interested in the mechanisms of aging and longevity. And as you can see on those nice self-portraits that Rembrandt painted of himself every year of his adult life, aging is accompanied by very obvious outward changes. But of course, it's accompanied by functional decline in all tissues. It's also accompanied by diminished resilience to a variety of stressors, susceptibility to not just one, but a constellation of diseases, and at the very end of life, generalized system failure and death. So just to give examples, if we take the brain as an example tissue, during aging, there is cognitive decline, working memory, short-term memory, long-term memory, and speed of processing all start declining pretty early, and this continues on. Aging, as I said, is associated with a striking increase in many diseases for which age is actually the prime risk factor, whether it's heart disease, cancer, type 2 diabetes, and of course, the exponential increase in the terrible neurodegenerative disease, such as Alzheimer's disease. Aging is not only accompanied by all those chronic diseases, but as we've seen this year, aging is linked to increased severity of infectious diseases and has been shown actually to be the primary risk factor also for the severity of COVID-19 this year. So really studying aging, the aging clock could perhaps give us cues for all those both chronic and severity to infectious diseases. For the longest time, aging was thought to just be the byproduct, the inexorable byproduct of wear and tear towards disease, decline, and death. But in the past 25, 30 years, it's been amazing to see the aging field showing that, in fact, mutation in simple genes could slow the rate of aging. Environmental factors could also slow the aging rate. And in fact, more recently, that some aspects of aging could in fact be reverted by blood factors from young animals, by reprogramming factors, and also by agents that kill senescent cells, for example. So in the next couple of slides, I will give examples of genetic and environmental factors that contribute to slowing aging and promoting health and longevity. So a great case study for a longevity gene is the insulin FOXO pathway, which was discovered in the worm C. elegans by Cynthia Kenyon, also Gary Rufkin, for regulating aging in worms, that has since been shown in flies, mice, and even human to be very important for the regulating the aging process. Notably in human, FOXO, which is downstream of this insulin pathway, is a quote unquote longevity transcription factors. And it's been shown that there are variants in humans in cohorts of centenarians in the US, in Europe, and also in Asia that are, in these cohorts of centenarians, variants of FOXO are associated with exceptional longevity. That's for an example for genetic regulation of lifespan by single genes, in fact. Of course, there are also environmental factors and compounds that have been shown to affect aging. This is just examples because the field has found many. Again, a case study is dietary restriction, restriction in food intake without malnutrition has been shown to extend both health span, the healthy portion of life without disease, and maximum lifespan. Exercise has been shown to increase health span, although the effect on maximal lifespans don't seem to be as strong as for dietary restrictions. And of course, there are compounds. Again, a case study is rapamycin, an mTOR inhibitor, one of the other genetic pathway that's very important for lifespan, that has been shown to have, in virtually all species tested so far, an extension, promote an extension of both health span and lifespan. Metformin, which activates many pathways in cells, including the AMP kinase, has also been found to extend to some degree both health span and lifespan. And some of those are in clinical trials currently. So this is a pretty exciting time for the aging field. In our lab, like many, were very interested about the mechanisms that convert a young, healthy, robust individual into an older, frail individual with increased chances of multiple diseases and also increased risk of death. And we're very interested in how those intrinsic mechanisms are impacted by environmental stimuli, whether it's food, diet, but also sexual interactions, pathogens, and whether this can give us new cues on how to slow, suspend, or even reverse aspects of this aging clock. Now, aging, longevity are very complex phenotypes. So to tackle this very complexity, what we've decided to use in our lab is a multi-organismal approach that takes advantage of respective strengths of different model organisms. So to discover new paradigm for the regulation of aging, we use models that are relatively short-lived and have a compressed lifespan to allow high throughput studies and discover new concepts for lifespan. So like many in the field, we use the worm C. elegans, which recapitulates an entire lifespan in a month. We've also pioneered this African killifish as a new vertebrate model for aging. It recapitulates an entire lifespan in about 4 to 6 months. But of course, to understand and translate principles of aging and rejuvenation in mammals, we use the mouse as a model and cells coming from mice and also from humans. And today, what I thought I would do is summarize recent work and also in the spirit of those kinds of seminars, present unpublished work in this realm of what we've done in mammals, in particular with regenerative stem cells. And then at the end of the talk, I will present our effort to develop the African killifish as a new model and the unpublished work we have in this area. So first, the stem cells. Why were we interested in regenerative stem cells? We felt that those cells had, in a way, figure out the longevity problem, in the sense that their lifespan of those stem cells can vastly exceed the lifespan of the organism in which they are, and they are present in a lot of adult tissue. So we were curious about two overall main questions. One, could preserving or maybe tapping into the potential of tissue stem cells be important to prolong tissue function during aging and perhaps even lifespan? And two, more conceptually, could studying stem cells, which have figured out how to live longer even than the organism that they are in, give us some cues at the molecular or cellular level of the mechanisms of quote unquote immortality or rejuvenation, just to understand a little bit better the cellular and molecular aspect. We are very interested in brain aging and rejuvenation. So we are interested in stem cells that are present in the brain. I know there's been some controversies in the field, but the human brain and the mouse brain has reservoir pockets of neural stem cells that are present in the hippocampus. And the main one, the one that's not controversial actually, is the ones where they are both present, very abundant in humans, in mice, in the subventricular zone. This is a sagittal brain section. So near the lateral ventricle, there is all this regenerative region. What's really cool is that the stem cells that are present there are normally pretty dormant, but under the right cues, they can activate, which means proliferate, enter the cell cycle. And they can give rise to all three differentiated cell types in the adult brain: neurons, astrocytes, and oligodendrocytes. So they are multipotent. And they can participate, they can actually, like their progeny can migrate, and they can participate to the neural circuits in other distal regions. So for example, they can migrate to the striatum in human, they can migrate to the olfactory bulb in both human and mice. They can also migrate to site of stroke or traumatic brain injury, and they've been shown to contribute to some aspects of learning, memory, olfactory discrimination, very important for COVID, actually, COVID-19, and repair upon traumatic brain injury or CTE, like the football players, and also repair upon stroke. So a key question is what happens to those stem cells which have this amazing potential during aging? And in fact, during aging, they lose two of their remarkable capacity. The first one is that they lose this ability to enter the cell cycle, and they remain very dormant, very quiescent. And then for the few that enter the cell cycle, they are actually less good at giving rise to the right proportion of neurons and astrocytes and oligodendrocytes, and they give rise to less neurons, as you can see on the left part of that graph. When we started the lab, just as a summary of published work, we were very interested in a candidate-based approach, and we asked the question of, can longevity genes, genes that were discovered because they affect organismal lifespan, could they preserve stem cells at the molecular and cellular level? And what we found is that this longevity, pro-longevity transcription factor, quote unquote, FOXO3, the ones that are present in centenarians, it is very important to preserve neural stem cells during aging. And we've identified, in fact, the targets of this transcription factor, and they are very important for metabolism and stress resistance. So that was very exciting to us, but it was also a candidate-based approach. So we were thinking like it would be really cool to have a more systematic view of what happens during the aging process, especially because the stem cells are present in this niche, in this what's called the regenerative neurostem cell or neurogenic niche, which contains many different cell types. In fact, actually probably more than 10 different cell types: the stem cells, the quiescent one, the activated one, their progeny, but also other cell types like oligodendrocytes, endothelial cells that form blood vessels. So it's really an excellent paradigm that in a very tiny piece of tissue, there is all these different cell types. And we wanted to know what are the age-dependent changes entire neurostem cell niche during aging. Are all the cells aging in the same pace? And can this knowledge be used to understand tissue aging and to rejuvenate, quote unquote, the tissue. So again, this is published work, but just to summarize, we've started by developing a fluorescence activated cell sorting protocol, a FACS protocol, by aging mice that have a transgenic marker that's present in stem cells. Suffice it to say that by combination of aging those cohort of genetically modified mice, in combination with 6 different antibodies, we've been able, this is based on Fiona Deutsch protocol in our stem cells, we've been able to isolate from the same animal 5 distinct populations of cells, neurostem cells, but also endothelial cell, and et cetera, and done RNA-seq at the time, bulk RNA-seq on them. And this was the work of a terrific graduate student in the lab, Dina Lehman. And what she found was that one aspect that changed during aging was a molecular signature for lysosomes, which are really important part of the protein homeostasis or protein quality control. And indeed, with staining, and this is just to summarize, we found that the quiescent, the dormant cells, which are the head of the lineage, have those gigantic lysosomes that actually go bad with age, and the protein aggregates start to accumulate. But we found that this can be reversed, at least to some degree, by nutrient deprivation that engages the lysosome, or by boosting the lysosome, and that this could ameliorate the ability of those quiescent neurostem cells, which are the head of the lineage, to give rise to progeny. So we've been very interested in those data, and we're currently pursuing them by trying to identify what are the protein aggregates that are forming in the neurostem cells. It's during normal aging, and what are their importance and their role in the aging of the neurogenic niche. More recently, we wanted to have not only a bulk view of the neurostem cell niche, and that we can purify the stem cells, but it's still in bulk. We wanted to have a single cell resolution of this niche. So another graduate, terrific MD PhD student in the lab, Ben Dolken, helped by Matt Buckley and Paloma Navarro, what they did together was to do single cell analysis from young animal and old animals, and to just have an unbiased view of all the cell in the neurostem cell niche during aging. And this is what's called the Tisney plot. It's a reduction in 2 dimensions of the single cell data, where each dot that you see on this graph is the full transcriptome of only one cell. And you can see that there are clusters of cells, and we recognize, by markers that there are microglia, oligodendrocytes, endothelial cells, the quiescent neurostem cells, of course, proliferative stem cells, neuroblasts, all the lineage. So this was cool to see. But with age, what was the single cell coming from old mice are in red, the single cells coming from young mice are in bluish color. And what we could see was that, of course, there were transcriptional changes during aging, and that's cool, and we're now investigating that. But the aspect that captivated us was we found that there was an infiltration in old neurogenic niches of cytotoxic CD8 positive T cells. And not only were they coming in the niche, those cells, but they were clonally expanded, meaning that they had the same T cell receptor. And this usually happens when they have recognized an antigen. So we published this study, and we still don't know what the antigen is, but where we are going with this effort is, do these T cells recognize brain-specific antigens then in old individuals? And what could those neoantigen in aged cells be? Could they be protein aggregates themselves that expose new peptides? Could it be reactivation of some viruses or transposons? Could it be new peptides leaking from mitochondria? We don't know, but we're very excited to find out. But today, also in the spirit of those types of seminars, I wanted to present our unpublished work also led by a terrific graduate student. We're very blessed with a terrific graduate student at Stanford. His name is Robin Yeo, and he asked, how do chromatin states change during aging in the regenerative neural stem cell niche? And could this tell us something new, actually, about aging and regeneration? Of course, chromatin changes are very linked to transcription changes, and we have done a lot with RNA sequencing that tests the transcription. But chromatin is also can be poised, it can be linked to different cell states. So we were very curious about maybe identifying something new with that type of approach. So we decided to use a technique that was actually developed at Stanford called ATAC-seq, which is a way to genome-wide probe the accessibility of chromatin, whether it's open or whether it's closed at the genome-wide level. And we can do that on purified cells. Now, we can do that even on single cells, but at the time that Robin started his PhD, that could be done on rare populations of cells that were purified. So he used the same fluorescence activating cell sorting technique that I mentioned, isolated endothelial cells that form the blood vessels in the niche, astrocytes that are in the niche, quiescent neural stem cells, which are very similar to astrocytes actually, activated proliferating neural stem cells, and then the more committed neural progenitor cells from the same mice, either young, 3 months old, that's the equivalent of a 20-year-old human, or old, 20 to 24 months, that's the equivalent of a 70-year-old human, or 75. So here is an example of a ATAC-seq peak, like how the data looks like, just at a specific locus for the neural lineage. It's called ASCL1. And what you can see is that ATAC-seq, like the way it works is that it's accessibility to what's called the transposase. So in fact, you see peaks at where the chromatin is actually open, which tends to be on regulatory regions, in promoters or in enhancer regions. So you get a lot of those regulatory regions on the genome. But what we wanted to know, this is just an example, is that globally, what does chromatin look like? Chromatin accessibility looks like, and in different cells and with different age. And what you could see is that the global chromatin accessibility is actually pretty amazing at distinguishing, as one could expect, actually, like cell types. So endothelial cells are in orange on the left part of this principal component analysis, again, a way of reducing the data in 2 dimensions. And then you have the neural lineage cells, the astrocytes, the quiescent neural stem cells, the activated neural stem cells, and the neural progenitors that are on the right of the graph. It's also pretty amazing at distinguishing cell state, whether a cell is quiescent or dormant, that's on the top, astrocytes and QNSC, or whether a cell is actually proliferating and active, that's at the bottom. But as you can see on the global, actually PCA age is actually not the main distinguishing factor. But when we zoom in to the quiescent and activated neural stem cells, we can see a very clear distinction with age in the PC axis number 3, also in the PC axis number 2, actually. And this was really neat because you can see that there is a difference by cell state. So the quiescent on the left in blue, the activated on the right in red, but there is this neat separation by age with the old at the bottom in circles and the young at the top with triangles. That was neat because in fact, when we had done RNA-seq with exactly the same mice, with exactly the same FACS protocol, RNA-seq was very good at separating the quiescent one as well, but not actually the activated cells, probably maybe because of poisoning, or maybe because of the fact that chromatin is analog, like there is 2 copies of changes compared to the vast range that you can get with RNA. But no matter what, this was encouraging because it told us, oh, maybe there is something different, slightly different about chromatin than there is with transcription, and that could lead us to new topics. So where are those chromatin changes during aging on the chromosomes, and what is their global changes? what was cool to see was that, in fact, so here you see the chromatin open is marked as a black tick mark, vertical tick black mark. And what you see lined up here are the different chromosomes of the mice. And what surprised us was that, in fact, there were opposing chromatin changes between quiescent and activated neurostem cells. And in fact, the quiescent neurostem cells had become more open in young and become more closed in old, less accessible in old quiescent neurostem cells. And it's the reverse for activated neurostem cells, that they are more close to begin with in young, but then they open during aging. So we were very curious about that and wondered, what are the genes that are underlying those opposing changes? Are they different genes for one type of cells and the other? Are they the same? So what are the genes and pathways then that change in old quiescent and activated neurostem cells during aging? Could this reveal pathways that we had missed with RNA-seq? When we did the Go-term analysis on those changes, we were excited to see that, in fact, these were new pathways that were showing up during aging in quiescent neurostem cells that we had missed before. The cell adhesion pathway, so negative regulation of cell mobility, CAMP-mediated signaling and response to force calling are also actually genes that are involved in cell adhesions. Those were more open in young and they closed in old in neurostem cells. And it was the same type of genes actually that underwent the exact opposing changes in old. So not the exact same signature, but still related cell signature. So adherence junction, cell adhesion that were, on the contrary, more open in old in the activated neurostem cells than in the quiescent one. So that was neat for us because in fact, when we re-examined the RNA-seq, we actually, both in single cell and bulk, we actually observed that as well, except we had missed it because it was lower on the rank compared to the lysosome changes and the inflammation changes, which were really at the top. But what that suggested that there is something really intrinsically fundamental, perhaps, about those cell state changes, chromatin linked to cell state changes with age. But we wanted to functionally test that. So what Robin did is that to functionally test this hypothesis was to obtain primary cultures from young and old brain and cultivate them for very limited passage and have them in quiescent state and the activated state, FACS sort them and do live cell imaging in them so that you can follow and track them. And you can calculate their velocity, you can calculate their different aspects, actually different metrics. What we could see, and this is represented by the fact that the y-axis have quite different numbers on them, is that to begin with, quiescent neurostem cells are more adherent in general than their activated proliferative counterpart. But then during aging, the effects are opposing, as was suggested by the chromatin, meaning that the quiescent cells, which are normally pretty adherent, become less adherent and actually more motile with age. Whereas by contrast, the activated neurostem cells, which are actually pretty motile cells to begin with, become less motile and more sticky, more adherent with age. This was done in a dish, so we wanted to verify in a more, not physiological, but in a context where there was more extracellular matrix, so, using the matrigel, and indeed we found that activated neurostem cells, which are usually pretty motile in the matrigels, are actually less motile with age. So that's all in vitro. So we wanted to know, does that also apply in vivo, where the cells are actually pretty stuck in the brain, at least in the niche? So we compared, and this is the work of a new MD PhD student, actually in the lab, Olivia Zhu. So she compared young and old neurostem cell niches and calculated the distance to the ventricle of the quiescent versus the dormant versus the proliferative neural stem cells. And what she found again was this opposing effect where the quiescent neural stem cells, they actually like, when they're old, are more distant, maybe migrated away from the ventricle where they're normally like really sticky there. And the activated neural stem cells, the ones that are actually more motile and supposed to actually be able to, in fact, leave the niche to give rise to progenitors that will populate repair region or even like the olfactory bulb, the striatum, like distal region, those are actually kind of stuck in the niche. And we also verified that by doing this time sagittal section and labeling the activated neural stem cells with EDU, which is a marker that it's an analog of a base, so it incorporates into cells that proliferate, so the activated proliferating neural stem cells. And what we could see is that those cells are highly present at the beginning in the niche and the rostral migratory stream, which is just after the niche, so that the cells normally go and then they reach the olfactory bulb after 2-7 days. So that's for the young animal. Of course, the old animal have less stem cells to begin with, but they seem to be more stuck also. So not only less stem cells, but they are more stuck in the niche. And that's kind of cool because it could also underlie some of the defects of the neurogenesis. It might not just be defect in cell proliferation, ability to enter the cell cycle, and skewing of the lineage towards neurons, but also the migration capacity to reach those distal regions of the brain in an old brain. So this suggested then that quiescent neural stem cells may become, that are normally very stuck lining the ventricle, they may become abnormally migratory with age, and that might, we don't know, but it's very speculative, but we speculate that this might actually be problematic for their normal function. Maybe those cells are considered the mother stem cells for glioblastoma, so the fact that they're more migratory could also like accentuate the bad features of the glioblastoma. Whereas in contrast, the neural stem cells, the activated neural stem cells and their progeny, the neuroblasts, do not migrate as well to the normal sites. And we have some preliminary data that might also be true for injury sites during aging, and that may contribute to the general brain defects during the aging process. So with this, then, we were curious, and this is work in progress, and it's unpublished work. So we were wondering then, can the migration defects in old activated neural stem cells and their progeny be restored then? So we leveraged the chromatin data and we asked, is there a hub when we use ingenuity pathway analysis? Like, what does it tell us about the possible handles that could be linked to those general changes in cell adhesion? And one hub that came from those unbiased analysis and also from reading the literature was the protein kinase named ROCK, which is essential to integrate vast numbers of cues and regulate. Of course, it's very pleiotropic. It regulates a number of aspects, but including cytoskeleton and cell shape and motility. And what's cool about ROCK is that there is a very nice, actually FDA-approved inhibitor called ROCK inhibitor Y27632. So we decided to use it. So far, we've only used it in vitro, and we're only starting to do in vivo experiment by injecting it. But we're very excited about this because in vitro, when we add the ROCK inhibitor to the old neural stem cells that normally has this reduction in their ability to migrate, this restores their ability to migrate through matrigel. This is a matrigel experiment. And also when we do the velocity assay at the single cell level, what we see is that it boosts, both in young and old actually, it boosts the ability of cells to really migrate intrinsically. So that's been neat for us. And overall, we are continuing our exploration. So this is like the work that we've done recently, but we are continuing our exploration of the aging, unbiased exploration of aging changes. Aging is accompanied by changes in the lysosome, protein aggregates, T-cell infiltration in the niche, and migration defects. So we'd like to understand more about those, whether they are linked or different. In the recent unpublished work, we were surprised that aging has in fact an opposing effect on quiescent and activated neural stem cells in terms of chromatin accessibility and in terms of cell migration. And we're curious about, of course, testing more the ROCK inhibitors in vivo, or maybe like that's more in the far future, could neural stem cells and our progeny be directed in vivo? Because here what's neat is that the change are cell intrinsic. So could we pilot them almost a little bit regardless of the extrinsic changes, which are also very important, but could we pilot the progeny by changing the chromatin to migrate to other sites for repair or injury of or degenerative diseases, maybe other regions of the brain even. So that's, again, highly speculative, but we're very intrigued by that possibility. All right, so now in the second shorter part of my talk, I will address our work to develop and use the African killifish as a new model to study aging and also quote unquote suspended animation. So what is this killifish? The African killifish lives, in fact, normally in ephemeral ponds, transient ponds in Zimbabwe and Mozambique that are only present during the rainy season, which lasts 4 months. And then the ponds completely dry up during the dry season, which lasts 8 months. So the fish has developed two amazing adaptations to survive this extreme loss of its habitat, which is water. The first is that's very helpful for aging research, is that it recapitulates an entire lifespan and life cycle very fast. It grows fast, it reproduces fast, and as a consequence, it ages fast. And that's really terrific for studying aging. The second remarkable adaptation is that 70% of its embryos and that's for the survival of the species, are able to enter a state of suspended animation, quote unquote, called embryonic diapause. And we're very excited about this state because during this state, the embryos are encased in this chorion. They're already very developed. They already have all major organs. They have a brain, they have a beating heart, they have muscle, and they can survive in this suspended animation state for months, even years. In fact, our record in the lab is 2.5 years. And when they come out, they don't seem, and we measure that, they don't seem to have trade-offs for growth, fertility, and even lifespan of the future generation. So that's a pretty cool, extreme biology type of feature that we are interested in studying. But for aging in particular, this is also super cool because it's the shortest lived vertebrate that can reproduce in captivity. It lives only 4 to 6 months. In this short life, it does display aging phenotype: fertility decline, muscle decline, sarcopenia, learning deficit, neoplastic cancer-like lesions. It has a hypothalamo-pituitary axis. It has blood cells, inflammation, inflammaging. It has stem cells in most of its adult tissues. So those are very exciting because those are features, important components that need to be taken into consideration for human aging. So it's a terrific model in that regard. So it lives short, it's a vertebrate, so this is cool, but it didn't have any tools for it. So the postdoc who really spearheaded this entire work in our lab is fantastic postdoc, Dario Valenzano. He now has his own lab at the Max Planck Institute in Germany, in Cologne. He actually knew about this fish because he worked on it as a graduate student in Italy with Sandro Celerino and learned about the biology of this fish. And he, in my lab, he decided to study it, to set up like the genomic platform that is needed to transform an interesting animal and species into an animal that can be studied in the lab. And he did that in collaboration with 3 fantastic other postdocs in the lab. Berenice Benayoun, she now has her own lab at USC. Parampriya Singh, he's still in the lab and is going to be on the job market, actually, terrific postdoc. And Itamar Harel, who now has his own lab at the Hebrew University of Jerusalem. So together what they did is that they sequenced the genome. That's what Berenice did at 2.2 gigabase. And Itamar, he set up a very nice CRISPR-Cas9, very versatile. It works really well, CRISPR-Cas9 editing pipeline. This work has been published. So for the CRISPR-Cas9, this is the work of Itamar. For the genome, this is the joint work of Dario, Berenice, and Param. Itamar also put out a protocol, and then Chico is another postdoc who just got a position at Stony Brook, actually, and has been working on genomics platform, but for studying more the diapose, the suspended animation aspects. So we are doing a lot in the lab, like to now develop more tools and also use these tools then to ask a number of questions, both about aging and suspended animation. For the unpublished work, again, in the spirit of those types of lectures, I thought I would talk to you about unpublished work of exploring the role of protein aggregates and prion-like behavior during aging using this fish. So this is spearheaded by Itamar Harel, again, a terrific postdoc, Parampriya Singh. And this has been a fantastic collaboration with a colleague at Stanford, Dan Yarosh, who is an expert in prion biology. And he trained with Sue Lindquist, actually, when at MIT, and Imbal Ziv Uzial, a postdoc in his lab, and Iwen Chen, a graduate student in Dan's lab. So protein aggregation, of course, very well known to be important and linked to a lot of age-related diseases in the brain. A-beta, Alzheimer's disease, tau, Alzheimer's disease, alpha-synuclein aggregate for Parkinson's disease, TDP43 aggregates for amyotrophic lateral sclerosis. And that's all extremely exciting and a very interesting field. But what we were wondering is, what about protein aggregates in the brain and actually other tissues during normal aging? Like, is it possible that in fact there are aggregates during normal aging? What are they? And could they perhaps contribute to disease? And do they form also in tissues other than the brain? So we took advantage of really a combination of features. One, the African killifish, and as a model. Two, quantitative mass spectrometry to have an unbiased view of protein aggregation. And three, the expertise of Dan Yarosh in prion-like biology. So we used a tandem mass spectrometry, which is called tandem mass tag, which goes in series of 10. So here the design is that we compared 3 young male fish, 3 old male fish, and 3 old male mutant fish for telomerase that we had generated with our CRISPR-Cas9 pipeline. And what's cool about telomerase mutant is that it's almost like a segmental progeria. They have age-related accelerated defect in some tissues in particular. We isolated 7 tissues: liver, brain, gut, testis, heart, muscle, and skin. Except we couldn't isolate testis for the TERT mutant, because in fact that's one of the tissue in which the degeneration, the age-dependent degeneration, is very, very pronounced, too pronounced to get tissue anymore. In each of those tissues, we used a protocol to isolate an aggregate fraction which has potential prion or prion-like properties based on molecular weight. And that's the work that originates from Dan Yarosh in actually yeast, where they compared all sorts of prions and determined that in fact, proteins with prion-like ability, they had a certain size where they were not yet into those very large aggregates like stress granules, but they were above the molecular size of molecular machinery, like the ribosome or the transcriptional machinery. So we did a native lysate and an ultracentrifugation on sucrose pad to really isolate a fraction based on high molecular weight. And we verified that indeed on silver stain, it contained like proteins that had aggregation properties and were SDS resistant. We then use tandem mass tag mass spectrometry. So that's a really neat way of having a comparative, a very quantifiable weights like Sci-like, but really like with heavy metals, basically to compare between all those different ages and across different samples. So lots and lots of mass spec. I think we had 120 samples in the end, because we have total lysate as a control, and then the protein with this aggregation propensity. We first verify the quality control for the data, and the proteomics data sets, especially with the tandem mass tag, are really amazing. So we were very excited about this, because usually for proteomics, it's always a little bit harder than genomics. So it was good, pleasing to see for us that the replicates have very good very good reproducibility between replicate 1, replicate 2, replicate 3 for tissue lysate aggregates. Even with principal component analysis, in most tissues, we were able to group based on full proteome or based on full aggregatome, if you wish, the aggregated fraction, the young samples from the old sample, from the old telomerase mutant sample. So a key question then is, what are the proteins that aggregate with age in each tissues? Are they shared within all tissues, or are they tissue specific? And we were very impressed to see that the protein that aggregate with age, there are some in every tissue actually, but they are, number one, very different and very specific. So the proteins that are aggregating with age in the brain are, you can see them in orange, they're lined up in this first block, and they're completely distinct from the one that aggregates in the gut, that are distinct from the ones that aggregate with age in the heart, and etc. And there are only a few proteins that are actually shared, and even not shared all across. And of course, part of that is due to tissue specificity of expression, but it's not just that, because we could see that the protein aggregatome was more tissue specific than the protein expression. And you have cases like this long chain fatty acid transporter, SLC27A1, where it's expressed at similar level in brain and heart, but only during brain aging does it aggregates, perhaps highlighting, and I'd be happy to talk about that in the Q&A, differential defects in the protein homeostasis network in different tissues during aging. But so old individuals exhibit increased protein aggregates in specific tissues during aging. But in the remaining part of the minutes of the talks, I'd like to present our work where we decided to delve deeper and in the brain and ask, are there previously uncharacterized protein aggregates in normal old brains? Because pretty much all the work has centered about diseased brain, but not normal old brain. And these are the top, from the left to the right, the top proteins with increased aggregation in old brain. And what was really cool to see is that there was an enrichment, which we confirmed by Go, for RNA binding proteins that aggregate in the brain, number one, and for proteins that had, and this was neat to see, and this is only in the brain that we saw that there was an enrichment for protein that had a prion-like score, even though, of course, we purified proteins based on their molecular weight to enrich for prion-like ability. But in the brain, they also have this high prion score. When it's positive, the more positive, the better. We particularly focused on this protein called DDX5, which has a very high prion-like score of 35. So DDX5 is an RNA helicase. It's an ATP-dependent RNA helicase, and it's involved in RNA splicing, notably the splicing of tau, actually, the normal splicing of tau, which is important, in fact, for Alzheimer's as well. So that was cool for us to see. And what was also a key feature is it had this very conserved prion-like domain in the C-terminal region, both in killifish and in human, that is detected by the prion algorithm and also by the disordered. So it's a disordered region detected by disopred. We verified that DDX5 not only was present in those fractions, but in fact, it's interesting, in both young killifish and in young mice, it's already in those nuclear speckles, and it's known actually for RNA binding proteins involved in in splicing, that they form those nuclear type of speckles that are probably the splicing machinery. But what was cool to see is that during aging, both in the killifish and in old mice, there was this kind of disorganization, more and then more cytoplasmic, puncta-like aggregates. So the conserved RNA helicase DDX5 exhibit increased aggregation in old killifish brain by biochemistry and tandem mass spectrometry, and aggregate-like puncta in old killifish and mouse brain. So finally, we were curious to test whether DDX5 could have prion-like aggregation seeding property. So in the interest of time, I'm just going to summarize in 2 slides the data that we have on this, but I'll just mention, like we have a bunch of data on that we're very excited about. So just in summary, we used this yeast assay that the Yarosh lab has used for protein aggregation, where you induce a protein in tagged with a red M-Ruby, red fluorescent protein. And if the protein has propensity to aggregate, you'll see puncta. And then you use the yeast system to completely dilute out these aggregates, except for seed, if there is any. And you ask, if there is expression at very low level of the same protein, but now in the green channel, is it going to seed the formation of this aggregate? And what was cool to see, just in summary, and I'll be happy to talk more in the Q&A session, is that indeed DDX5 is able to aggregate, and there is a number of other proteins that we've identified in the brain that can also aggregate and form those puncta in the yeast assay. But what was really cool and actually pretty unique about DDX5 is the fact that it can still form some aggregate in the green channel, even after dilution of the protein, suggesting that it has this seeding property, and that we found that this was very dependent on the C-terminal prion-like domain. We've also found that DDX5 has other characteristics of prions. It has species barrier, it forms immobiles aggregate, and recently we found that it can form those condensate, so phase-separated condensate. So we're also very interested in that and understanding how this changes with age. So to summarize this part, we found protein, this second part, protein aggregates form in 7 tissues during aging in vertebrate model. In the brain, the DDX5 helicase aggregates with age and exhibit prion-like properties. And we've been curious about the effect of DDX5 on brain function and potential spreading of cognitive defects. So I know it's 12:53, so I can stop here and go to the acknowledgement, but I have a few slides on cognitive function in the killifish. So what do you think, Dr. Hoddes? Should I?
Yeah, why don't you go ahead? That'd be very interesting to see the functional cognitive chordates here.
Okay, cool. Yeah, so it's just like a very brief, how can we measure behavior in the killifish? So we've embarked on this project first with Andrew McKay, but now we are doing more with actually Carl Disseroth here at Stanford with a transformative R1, nicely supported by the NIA and TR1, so that's really nice. What Andrew is a graduate student at Stanford with engineering capability, and he's designed an automatic feeder for the African killifish. So we can feed 200, 400 tanks at once. So that's really cool for feeding, and this is really helpful also for drug delivery, and we're investigating that. But for this particular context, we've decided to pair it with a stimulus so that we can associate the food with a light, a red light. So that's really neat because the red light, we can program it, it's fully programmable. So the red light turns on, it waits 3 seconds, and then food gets dropped automatically. And you can see how the fish can learn to associate this stimuli. So for example, here it's before training, you have the red light, the fish doesn't care before training, and it's 3 seconds, the food will drop in the tank, and then it sees the food. It's hard to see the food itself, but the fish sees it and it goes at the top and it eats it. So it doesn't care about the light because it hasn't yet associated the light with the incoming presence of food. But the fish are really smart. So after training, like they do a beeline because they know that as soon as the red light comes, and they even like here, they beat in the water because they're waiting for the food to drop the 3 seconds. And then the food finally drops and then they start eating it. But this is a really neat assay because it allows us basically to calculate trajectories with a machine learning, to train a machine learning algorithm with deep lab cut, and to have, before training, the fish don't really care when the red light, they see it, we verify that they see it, but they don't really care about it. But after training, they make this beeline to the top because they know that food is coming. And we can then use that to calculate a learning index. And it was neat to see that, as many things, the cognitive or learning index declined with it. But we're very excited about this because this is a high throughput system. So the automatic feeder allows scalable lifespan, cognitive function studies. This can be done longitudinally. So we are with 2 new postdocs, Claire and Ravi, we are trying to film the fish, to video record the fish during their entire life and see what behavior could be predictive of impending death or even long time death. We want to screen now that we have those automatic feeders for drugs that impact lifespan. And because, of course, the fish is really nicely genetically modifiable, to test genes that are implicated in disease and maybe do screens for like the genes times the compounds or environmental different stimuli. So I'll stop there. And I would really like to thank the people the terrific team. I've thanked them along the way, but here they all are. Our terrific collaborators at Stanford, Tom Rando, Tony Wiscori, Peggy Godell at Baylor, sorry, made a mistake in her name, and then Carl Disseroth and Danya Roche at Stanford. And my gratitude to the National Institute of Aging that has supported our lab for now 17 years. And I'm very, very immensely grateful for the support. So thank you very much for your time, and I'd be happy to take questions.
Thank you, Anne. So we do have some questions. The first noted that nutrient deprivation boosts old quiescent neural stem cells. Do you think this is due to the activated autophagy process? Obviously, autophagy is very central to a lot you've talked about, including protein aggregates, even prions.
Yes, so we think that that's likely a large part of that because when we expressed, for example, we saw that by manipulating mTOR, which is also very important for autophagy, we did also rescue some of the effects. We also were able to overexpress the TFEB transcription factor, which is involved in lysosome formation and downstream with autophagy. But we also think that there is probably a very lysosome effect that's in part dependent on autophagy, but also just the lysosome itself is having already a big impact on clearing a lot of the defects. Yeah.
A couple of questions, including one of my own, about any relationship that you want to speculate upon for cellular senescence, presence or absence, relevant or not to the stem cells that you've been focusing on.
Yes, that's another terrific question. We have used, like many others, we were using the P16 mice, and with the FACS system, we didn't see a lot, the P16 mice from Jan van Dersen and also the ones from Judy Campisi. When using the FACS, we didn't see actually a lot of senescent, or P16 positive, I should say, cells in this regenerative niche. But I also think that this needs to be reinvestigated because I think with FACS, we actually probably de-enrich for senescent cells, and this needs to be all reinvestigated by the new technology and transcriptomic. And we actually have submitted a consortium like CENNET, to reinvestigate those types of questions. I think it's a terrific question.
Great, glad you're interested there too. Any clues that might have come from your transcriptomics or ATAC-seq, and where you might have mined for any suggestion of senescence from those patterns?
Yeah, so there again, we didn't see as much as we were anticipated, but I also feel like same, even for single cell chromatin, they all go through this like tissue extraction
Yeah.
And even though some of it is without facts, I think also maybe the very fact of dissociating the tissue, and especially it's a very fragile region of the brain, we may have definitely enriched or lost them. So I think that's why we want to use, like CyTOF or MIBI and like those new like techniques which are in situ, so that we can unbiasedly, but in the tissue, assess them.
Great. And as Anna's commenting, there's a Common Fund initiative. I'm glad she's among those who responded to, trying to overcome some of the constraints, inflammatory context in the brain as you've looked at it.
Yeah, and that may actually also be directly linked to the SASP and senescence. Like, what comes up very, very strongly are inflammatory aspects. But so especially interferon gamma, which is secreted by T cells, was one of the main signature. And we are trying to collaborate actually with Chris Garcia here at Stanford, who does a lot of protein engineering to really like either block specifically the T cells, but also to, manipulate the cytokines, the different types of cytokines, because we've come to realize that some of them are anti-inflammatory and some of them are pro-inflammatory. So we'd like to test them individually. Yeah, that's another terrific question. Thank you.
Maybe another very broad question. The advantage of the multiple species that you've looked at with varying lifespans. So presumably aging and the changes you see if you crosswalk is proportionate to the lifespans somehow. So of course, a huge question is what is regulating the pace of aging across these species? Care to speculate?
Yeah, we are so curious about this. Like we've been deriving machine learning based clocks in the different species to using the single cell data set actually to really like try to understand that. Of course, the clocks, they may not tell us about the driver, but what I can tell you is that the clocks we've derived, a lot of it is inflammatory actually. So one of the speculation we had was that maybe like so it's true that a lot of people think about inflammaging as more like a later consequence, but we speculated based on those clocks that maybe we may not see it in a younger or 30-year-old or 35-year-old, but they might already be, changes in the immune system that might actually like already predict basically an different aging trajectory. So, but I know that it's usually considered a later predictor, and it would be nice to do causative, of course, studies like the clock is, a readout more. Yeah.
Yes, and I'm sure you'll continue with looking at what interventions, environmental and other, can alter the course of these. Well, I think we're a little bit past time. I think we should thank you. Regrets that we couldn't all be with you in person, but a large audience had a chance to hear from you, be inspired and informed, and we'll say farewell to the larger group. And for those who are fortunate enough to have a place with you at the tea at 4:15, look forward to it then. So thank you, Anna. Spectacular talk and work.
Thank you so much, Michele and all.